Welcome to today's lecture on cell biology. In this session, we will delve into the fascinating field of molecular biology and explore the intricate mechanisms behind some of the most important techniques used in genetic engineering and biotechnology. Our journey will take us through topics such as restriction enzymes, DNA ligase, plasmid DNA, transformation, polymerase chain reaction, PCR, gel electrophoresis, DNA sequencing, recombinant DNA technology, CRISPR/Cas9 gene editing, protein expression systems, transgenic organisms, and their applications. In this section, we will discuss the importance of restriction enzymes in genetic engineering. These proteins play a crucial role in identifying specific DNA sequences and cutting the DNA at those locations. This enables the creation of fragments with cohesive or blunt ends. Cohesive ends can join with compatible ends, aiding in the insertion of foreign DNA into plasmid vectors. Blunt ends, on the other hand, are unable to form hydrogen bonds, making them useful for certain applications. In this section, we will explore the pivotal role played by DNA ligase in DNA joining. As an enzyme, DNA ligase connects DNA strands by forming phosphodiester bonds between adjacent nucleotides. This vital function ensures the seamless sealing of any breaks or gaps in DNA molecules, preserving their integrity. Particularly in DNA cloning, DNA ligase is leveraged to fuse a foreign DNA fragment with a plasmid vector, leading to the creation of a recombinant DNA molecule. Next, let's delve into the world of plasmid DNA. Plasmids are small, circular DNA molecules found in bacteria and other organisms. They can replicate independently from the host genome and carry genes that provide advantages to the host cell, such as antibiotic resistance. In genetic engineering, plasmids serve as essential tools for the insertion, amplification, and expression of foreign DNA fragments. Researchers can manipulate plasmids to include specific DNA sequences, making them ideal for the production of recombinant proteins or the study of gene function. Now, let's turn our attention to transformation and bacterial hosts in cloning. Transformation is a process by which foreign DNA is introduced into bacterial cells. Bacterial hosts, such as Escherichia coli, are commonly used for this purpose due to their ease of cultivation and well-understood genetics. Transformation can be achieved through various methods, including heat shock, electroporation, and chemical-mediated uptake. Once inside the host cell, the recombinant DNA can be replicated and expressed, allowing for further manipulation and analysis. In genetic engineering, selectable markers help identify cells that have taken up desired DNA. Plasmid vectors are DNA molecules that carry foreign DNA and replicate independently. They enable efficient propagation and expression of recombinant DNA in host cells. These tools are vital for molecular cloning. Let's now explore the revolutionary technique known as the polymerase chain reaction, PCR. PCR is a powerful method used to amplify specific segments of DNA in vitro. It involves a series of temperature cycles that facilitate DNA denaturation, primer annealing, and polymerase-mediated DNA synthesis. PCR has numerous applications in research, diagnostics, forensics, and genetic testing. It allows for the rapid and accurate amplification of DNA regions of interest, even from minuscule amounts of starting material. Now, let's delve into the fascinating world of gel electrophoresis and DNA fragment analysis. This powerful technique allows us to separate and observe DNA fragments based on their size and charge. By loading DNA samples into wells of a gel and applying an electric field, smaller fragments move at a faster pace than the larger ones. By comparing the migration patterns with known standards, scientists can determine fragment sizes and explore genetic variations. Next, we explore DNA sequencing techniques. DNA sequencing is the process of determining the precise order of nucleotides in a DNA molecule. There are various methods and technologies available for DNA sequencing, including Sanger sequencing, next-generation sequencing, NGS, and single-molecule real-time, SMRT, sequencing. These techniques have revolutionized genomics and have allowed for the sequencing of entire genomes, enabling insights into genetic diversity, identifying disease-causing mutations, and understanding evolutionary relationships. Recombinant DNA technology encompasses a wide range of methods and strategies used to manipulate DNA molecules and create genetically modified organisms. It involves the transfer of DNA between different organisms or the modification of existing DNA within an organism. Recombinant DNA technology has revolutionized the fields of medicine, agriculture, and environmental science. It has enabled the production of life-saving drugs, the development of disease-resistant crops, and the study of gene function and regulation. Now, let's delve into the exciting world of CRISPR/Cas9 gene editing techniques. CRISPR, clustered regularly interspaced short palindromic repeats, and Cas9, CRISPR-associated protein 9, are components of a bacterial defense system against viral infections. Researchers have harnessed this system to precisely edit genes in various organisms, including humans. The CRISPR/Cas9 technique offers simplicity, efficiency, and versatility in gene editing and has revolutionized the field of genetics, paving the way for potential applications in gene therapy, disease modeling, and biotechnology. Moving forward, let's explore protein expression systems and recombinant protein production. Protein expression systems are used to produce large quantities of specific proteins for research, therapeutic, or industrial purposes. These systems can be based on bacteria, yeast, insect cells, mammalian cells, or cell-free systems. Through genetic manipulation, the target gene is introduced into the expression host, which then synthesizes and assembles the recombinant protein. Recombinant proteins find applications in various fields, including medicine, agriculture, and biotechnology. Lastly, let's discuss the creation and uses of transgenic organisms. Transgenic organisms are those that have had foreign genes inserted into their genome, resulting in the expression of new traits. Transgenic technology has revolutionized agriculture, allowing for the development of crops with enhanced nutritional value, resistance to pests and diseases, or improved tolerance to environmental conditions. Moreover, transgenic animals serve as important models for studying human diseases, enabling the investigation of gene function and potential therapeutic targets. In conclusion, molecular biology has allowed us to devise effective methods for altering DNA and investigating cellular functions. Techniques such as DNA sequencing, CRISPR/Cas9 gene editing, and protein expression systems have significantly impacted medicine, biotechnology, and genetic research.